Hi everyone, my name is Nan Jiang, a postdoc in Dr. Eric Goodworth's lab at The Ohio State University. Today I'm going to share with my interesting research about how pigments move in plants. So pigments not only have a well-known function and role as giving plant cell a blue or purple color for attracting insects to flowers, but also protect the plant cells but also protect the leaves against the damage from ultraviolet light. So we know the function of pigments pretty much, but how to transfer them inside the plant cell and keep them working well in plant is not well known. So imagine a plant cell is a factory. It's a factory for producing all kinds of stuff, including pigments. After generating them at the working station, the pigment needs to be packed up and moved to warehouse for storage. So my research is trying to understand how pigments are transported from the site of the synthesis to the site for storage. When I'm thinking about transportation, a question came to my mind is, do pigments move along inside the cell or by themselves? Or they need other partners during the trafficking. So after analyze the purified pigments from plants, so we know from the plants, the study found out that pigments require a protein called the TT19 as a protector to prevent them from degradation during the process. If TT19 is mutated or dysfunctional, the pigments in plants will not be accumulated and the subsequent loss of the color, say in the flowers. Now we know the pigments need a right and do not move along. So my following questions are how TT19 binds and protects pigments, and is TT19 the only the pigment binding protein, or there are other the unknown partners? To answer those questions, I use the forward genetic screening and the next generation sequencing approaches to try to identify the proteins that are functional related to TT19 during the pigment accumulation process. During the accumulation process, the result indicates some proteins I found have an opposite function as TT19 suppressor, which means mutated those proteins can restore pigment accumulation, although TT19 is dysfunctional. Now I'm trying to interpret the data and make sense about interaction between the pigments and the TT19 and then those TT19 suppressors. For take-home message is the accumulation of pigments in plants require other proteins to prevent them from degradation during the trafficking from the site of the synthesis to the place for storage. Thanks for your attention. Thanks.